guys. This video is sort of a continuation of what we started last week. It's just a little bit about the cell membrane's structure and then a tiny bit about its function, which we'll finish up talking about its function at the end of this week. But this goes right along with the activity that you guys did with your lab groups. I just wanted to kind of mention a few things about why we care about the cell membrane and also just some other stuff related to its structure in video form. So first of all, before I start talking about the cell membrane, it's really important to mention that surface area and volume are really important to cells. So for cells, the goal is that there is a large surface area to volume ratio, um, basically meaning that there is a bigger surface area than there is volume inside the cell. So I'm just going to make like a note here that says large surface area is ideal, which is kind of also mentioned here. So because of this, the size of cells is limited to being very small. Because what happens is the cell membrane is responsible for allowing things like nutrients into the cell, nutrients, oxygen, water, and removing waste. If you have like a giant cell with a ton of volume, it needs more nutrients in order to survive, but it doesn't have the surface area to absorb those nutrients. So a cell that's large, that is that large, isn't gonna do very well because it's gonna take so long for any sort of nutrient or oxygen or anything to get um, to the organelles inside the cell. So cells, generally speaking, will have a large surface area because they'll have more membrane than they have cytoplasm, which is the volume inside of the cell. Some examples of cells that have like super huge surface area to volume ratios are plant root hair cells and also microvilli, which I'll show you some pictures of those things right now. So here's a plant root. This is a super zoomed in picture. Um, and the cells of roots are actually called root hairs, root hair cells. They're not really hairs, but they look like them. So that's why. And if you've ever grown a plant where you could like see the roots, you actually can kind of see that. It's better with a microscope, but you can see like how they branch. The purpose is to absorb water from the soil or the surroundings of the plant because plants only get their water from their roots. So you can see here in this diagram that the surface area, the amount of membrane you have, which is going to be like all of this. It's a really big highlighter. I'm going to try that again with less size on that. Let's try this. So you have all of this, this, and then all of this. So you have tons of area where material can diffuse into the cell, most importantly, water. And you don't have that much volume compared to that has to be filled. So that's one example. The other example for us is in your small intestines. You have intestinal cells, and then they're called epithelial cells. So they're down here. This picture is pretty blurry, but I'll talk about it anyways. So these little like folds in the membrane are called them, oops, everything is so large today. So these are called microvilli. And what they look like is literally just tiny folds in the membrane of the cell. So all of this would be cell membrane. And it makes it so that all of these folds have the ability to absorb nutrients from the food that you ate. So your small intestine is actually the most absorbent part of your digestive system, where most of the nutrients will actually be incorporated into the cells there. So the microvilli are really important for allowing that to happen and giving them a super big surface area. And then here's like a more zoomed out picture of that. But there you go. Okay, so that's kind of why it's important. But now let's talk about the actual structure of the cell membrane. So the cell membrane, probably you've heard this term before, is also called the plasma membrane or the phospholipid bilayer. So you can call it any of those things. I'll just put a note up here. Lipid bilayer. Only two words, but two long words. We call the phospholipid bilayer selectively permeable. And what that means is that only certain things are able to pass through it. So I'll just say only some molecules can flow through membrane. Permeable means like able to pass through. And then obviously selective means only some. Most of the membrane is made up of phospholipids, which we talked about that last week when you guys did the activity, but they're the things that look like the little jellyfish. The head is phosphate and the tails are the fatty acids. We call things that have both hydrophilic and hydrophobic parts, or polar and nonpolar parts, amphipathic. Sorry, hard to say. Amphipathic. So it means that it has the properties of nonpolar and polar things in its different regions. So for my phospholipid that I just drew, the tails are fatty acids, which are nonpolar and hydrophobic. The head is a phosphate group. So back to functional groups. It's polar and hydrophilic. So the whole thing would be called amphipathic because it has both of those parts, hydrophobic and hydrophilic. Just like you guys talked about in your activity with your lab group, these phospholipids form bilayers because the phosphates want to be by water and the tails don't. So the tails sort of attract one another and face inwards to make this sort of double layer of phospholipids surrounding your cell, because most of what's inside of your cells is water and most of what's outside of your cells is water. Just kind of an interesting thing that originally scientists thought that proteins coated both the inner and outer surfaces of the cell membrane. Now we know actually that proteins pass through the membrane, which I'll show you a picture of that in a second. But originally, scientists thought that the proteins were just kind of sitting on the outside. And there are some proteins that sit on the outside, but there are also some that are able to pass through. So those proteins that are passing through the membrane, we would also consider them to be amphipathic because they have both hydrophilic and hydrophobic regions as well, where the hydrophobic region would be in the middle of the tails, the fatty acids, and the hydrophilic regions would be facing the outsides of the cell by where the phosphates are. Okay, here is just sort of a photo of your phospholipids, forming a bilayer because it's double layer. And then a very zoomed in version that just shows you like here's the fatty acids, tails, and here's your phosphates, phosphate group. And that's what forms your cell membrane. As this is like moving around your cell, it is moving. So the phospholipids actually are able to like swap places with each other side to side and even swap places with each other across the membrane. So just kind of something to keep in mind that this is constantly in motion. It's not like a wall. It's like being in a lake where there's like a bunch of buoys and the buoys are like kind of moving in the lake. It's and not like a brick wall or a dam. So just to kind of visualize. All right, so along with that, makes sense now to talk about the fluid mosaic model, which is a way to describe the cell membrane's fluidity. And like I just said, the phospholipids are able to shift past each other side to side or laterally, and then more rarely flip across the membrane. That doesn't happen quite as often. This can happen because the phospholipids that are next to each other aren't like covalently bonded together or anything like that. They're just experiencing sort of weak interactions that are keeping them in place. And then proteins are also able to kind of move through the um, the matrix of the membrane, and they're usually controlled by the cytoskeleton, which is a bunch of like filaments that are on the inside of the cell. We haven't talked about the cytoskeleton too much, but it's a bunch of like long filaments of microtubules that are able to kind of anchor the proteins and kind of control their movement. So again, you could think about this too as like if you have a swimming pool and you take the contents of a ball pit that you used to play in like at McDonald's or whatever, dump the ball pit into the swimming pool, that would be like the tops of the phosphate groups, the phospholipids floating on the top. And then imagine you also put into the pool some like large floats. Those would be like the proteins. So everything is still able to move and is moving and is shifting around, but it's all like in the pool together. So hopefully that helps with the image. Also, in the membrane is this very important friend who gets a bad reputation. Oops, called cholesterol, um, which helps with the rigidity and stability of the membrane. So cholesterol is actually really important for the cell membrane because if you don't have enough cholesterol, your membrane is like way too fluid to the point where like things are able to escape from in between the phospholipids. Um, and also, it's helpful in making sure that the um, membrane doesn't freeze if it's low temperature. So basically, it just helps the membrane maintain stability in terms of its fluidity, and it keeps it from becoming either too rigid or too fluid. So it's actually really important to have that cholesterol. Obviously, cholesterol also can be super bad, but in this case, it's really helpful to your cells. And then the last thing that's also part of the membrane is glycoproteins and glycolipids, which you might remember them from like a short slide in biochem, but they're basically sugars that are attached to either a lipid or a protein. So this glyco is sugar. So sugar plus protein equals glycoprotein. Sugar plus lipid equals glycolipid. They would be covalently bonded. So the sugar would be covalently bonded to the protein. These are really important for your body being able to recognize both that your cells belong to you, that they're supposed to be in your body, and also that your cells don't belong to you, or that other cells don't belong to you and they're foreign, like invaders. Um, they're sort of like the signal to your body that a cell either belongs there or doesn't. So just to kind of summarize this fluid mosaic model, this top part, the phospholipid shifting around, is what makes it fluid. All the parts sort of like combine together, the proteins, the glycoproteins, the glycolipids, the cholesterol. That's what makes it a mosaic, which if you've ever experienced seeing a mosaic or making one, you know that it's just like many different pieces together that come together to make a picture. Um, and that's sort of what I was trying to describe with the like ball pit and swimming pool with the floats and all that stuff. That would be kind of like a mosaic. So that's why it's called that. Here is an image that shows what's happening here. So you have the bottom of this picture is inside the cell. Inside cell. This is outside cell. which we call extracellular. So this word right here, extracellular. And inside the cell is intracellular, or in this case, they put cytoplasmic. You could also see intra. These like rope looking things on the inside of the cell are the cytoskeleton that I was mentioning. So you can see that it's kind of just anchoring things into place and preventing everything from sort of drifting away. The extracellular area also has some fibers. They wouldn't be called the cytoskeleton, but they're out there too, just kind of making sure that it's not going to like float away. So you can see here that you have your phospholipids making the bilayer. They go all the way back, and they would come all the way forward too. So it's making like a ball of this. It doesn't have to be circular, but whatever the shape of the cell is, it's 3D. And you can see sort of why I was describing it as a ball pit, because the phospholipids are kind of looking like that. Then you have different proteins. So you have your integral proteins that pass through the membrane. This is going to be on the next slide, so I'm not going to write it. And then peripheral proteins that sit either on the inside or the outside. These ones are sitting on the inside, but they could be sitting on the outside as well. Then you also have here your like little glucose chains that are attaching to this lipid. So this would be a glycolipid. And you also have over here a glycoprotein, where this chain of sugars is stuck onto this purple protein. And again, those would be used mostly for cell signaling and cell recognition. So that's usually why they're pointed towards the outside of the cell, so that other cells can recognize them and see what they're looking at. But just remember that this whole thing would be constantly in motion, floating, wavy, wiggly, shifting, all the time. All right, so now to talk about these membrane proteins, there's two different types. Your integral proteins are the ones that are stuck all the way in the membrane. They could also be only partially stuck in the membrane. So if they go, I'm going to draw just like a little section, a very simplified one. So here's some phospholipids. I'll put a protein right there. And then I'll put one that goes not the whole way. All right, so this is a very bad cell membrane, but let's say you have a protein here. This guy would be considered integral. This guy that only goes halfway would still be considered integral. So both of these are integral proteins. They can be hydrophilic, or they can be hydrophobic, or they can be amphipathic, which would be both. A lot of times these integral proteins are channel proteins. So I'll put like a channel here. Or transport pumps, which we'll talk about them towards the end of the week, but we'll call this a channel. And that's going to allow things that like normally couldn't pass through the membrane, because remember, selectively permeable, to be able to pass through the membrane. So let's say something is like charged or polar, or just like very large, might use a transport protein to pass through those fatty acid tails and get into or out of the cell. So they don't have to be channel proteins, but often they are, and we'll talk about them later on. Channel proteins don't usually use energy, but a protein pump or a carrier protein might use energy to move things against their concentration gradient, which again, we'll talk about later on. And then you also can have peripheral proteins. So I'll put one on here in pink. So a peripheral protein just sort of sits here on the top, very loosely bound to the cell membrane. And they might also be like a glycoprotein, but they're just hanging out here on the top. So this guy would be peripheral. They could also be on the inside, but either way, they're just like, they're not embedded at all in the membrane. And your peripheral vision is like on the sides. So just remember, like, this is on the sides of the membrane. Okay, cool. So this just kind of shows you, again, another image of the bilayer, but this time with proteins embedded. All of these would be integral proteins, it looks like. I think that one towards the back is also embedded in the membrane. Um, and then just the recognition that the parts that face outside or inside and are near the phosphate heads would be hydrophilic polar regions, and the middle would be hydrophobic. So this is also another reason why it's really important that proteins fold correctly and that the correct amino acids end up in the correct order. Because if they don't, your membrane protein could fold wrong. And if it folds wrong, you will not be able to stick your protein into the center of the membrane, which means that you might not be able to transport something or communicate something or function properly. So this is just another reminder of why proteins shape and why the exact right amino acids get put in the exact right order, which that's coded for by your specific DNA. So just a reminder of like, ding ding, very important. Okay, almost to the end, but these are just some important membrane functions, which we will talk much more about um, as we get to our lab, especially this first one, transport. So the purpose of transporting things into and out of the membrane is to maintain homeostasis. And if I had to say like what the main function of the cell membrane was, it would be this, maintaining homeostasis. Because if you don't maintain homeostasis, you're dead, quite literally. These transport situations include diffusion, osmosis, active transport, and then also like the cell eating things, which is called endocytosis, or removing things, which is called exocytosis. Um, and we'll talk more about each of those things later on. But there are also sort of these secondary and not necessarily less important functions of the membrane, which include acting as enzymes. So proteins, enzymes are proteins, but they are embedded in membranes and they are responsible for some major processes that your body needs to stay alive. One main one is ATP synthase, which produces ATP. If you forgot, ATP is energy. We'll talk about that later on when we get to cell respiration. But if your body couldn't produce ATP using this membrane protein, you would not, you would cease to function. um, Also, the membrane is really important for signal transduction, which means that when the cell receives a signal, there's a receptor protein on the membrane that accepts that specific signal and then starts basically like a really speedy cascade of signaling to other proteins throughout your cells to be like bing, 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 almost like really fast and efficient telephone to tell a specific part of a cell to make something or stop making something. Um, and that starts with the membrane being able to receive that signal at first. Also, membranes are responsible for linking together proteins. The structure is called a desmosome, one of the structures, but it just allows cells to adhere to one another really tightly. This is really important for things like your skin. It's obviously crucial that to create that protective barrier, that's the first like line of defense of your body, that the cells are really, really, really tightly bound. And there are proteins on the membranes that are responsible for creating those really tight adhesions between the cells. If you didn't, your skin would not be as good of a barrier or as good as keeping things in your body that you want to keep in. Finally, the last function of the membrane is recognition. So I talked kind of about the glycoproteins being able to um, serve as indicators that cells are where they're supposed to be or doing what they're supposed to be doing. One example of this is your blood type. So the part of your blood type that's A, B, or O. That is a signifier of which glycoproteins are found on the surface of your red blood cells. So if you're type A, you have A glycoproteins. If you're type B, you have B glycoproteins. If you're type AB, you have both types. And if you're O, you have no glycoprotein. So this is the reason why, if you're type O blood, you are considered to be the universal donor, specifically O negative, which we'll talk about the negatives later on, but because you have no glycoproteins on your cells. So that means that type O blood can be given to anyone because their immune system won't recognize it. They're like stealth mode cells because they don't have any glycoproteins. So they can just kind of like enter into the body and be stealth mode without triggering the immune system to like go into septic shock. Um, type AB can only receive, I'm sorry, type AB can receive any blood type because they have all the glycoproteins. So they can receive type AB, they can receive type A, or they can receive type B because their immune system recognizes that all of those glycoproteins are already found in their own bodies. But type AB people can only give to other type AB people. So that's just one example of glycoproteins. There are more, but that's the most common, easiest to understand one. All right. And here's just some pictures that are kind of showing that going on. Transport is happening here. So you can see some of it requires energy, some of it doesn't. We'll talk about that later on. Enzymes sort of catalyzing reactions, could be breaking stuff down, could be producing something. Here's a signaling molecule that's being received by the cell, and it's going to start sending that message throughout the whole cell, like telephone. Here's a cell recognizing another cell using the glycoproteins. So it could be recognizing that it's supposed to be there. It could be recognizing that it's a cancer cell. It could be recognizing that it's a foreign invader. Here's an example of two proteins joining a cell really closely together with another. And then also the proteins can be used to attach either to the extracellular matrix or to the cytoskeleton on the inside. So those are just a few of the main membrane functions, which we'll dive into each of these membrane functions individually kind of throughout the year. We won't do all of them in this unit, but we will do transport. So that'll be our next thing. Great job. Thanks for watching. You're killing it. See you soon.